on His Majesty's service. Receptors. Receptors, or sensory nerve endings, are specialized terminal structures of dendrites, often accompanied by glia and cells of connective tissue, which together provide the transformation of stimuli from the external or internal environment into a nerve impulse. In some receptors, for example taste and auditory ones, the stimuli are directly perceived by specialized cells of epithelial origin which do not generate nerve impulses but act on the dendrites that innervate them, changing the secretion of the transmitter. There are several classifications of receptors, depending on the location in the body. Exteroceptors are located near the surface of the body, in the skin or mucous membranes, and perceive stimuli from the external environment. Interoceptors are located in internal organs and perceive internal stimuli. Proprioceptors are receptors of the musculoskeletal system which perceive stimuli from muscles, tendons, and joints. They are a special kind of interoceptors. Depending on an adequate stimulus. Osmoreceptors perceive changes in the osmotic concentration of liquid, usually in the internal environment. Photoreceptors perceive light. Thermoreceptors perceive temperature. Chemoreceptors sense the effects of dissolved or volatile chemicals. Mechanoreceptors perceive mechanical stimuli, touch, pressure, stretching, vibration. And pain receptors or nociceptors perceive stimuli, chemical, thermal, or mechanical damaging factors which lead to pain. Depending on their structure, free nerve endings consisting only of the terminal branches of the dendrites, and encapsulated nerve endings, which in addition to the dendrite contain neuroglial cells surrounded by a connective tissue capsule. Exteroception is the perception of stimuli originating outside the body. They are detected by exteroceptors and include touch, pressure, heat, cold, pain, vision, hearing, smell, and taste. Being our largest organ, the skin possesses a wide variety of exteroceptors, including both free and encapsulated nerve endings. All of them work in the same way. The applied stimuli deform, compress, or otherwise change them. It leads to the generation of the so-called receptor potential in the membrane of the unmyelinated terminal portion of dendrite. Ion channels responsible for receptor potentials may be mechanosensitive, thermosensitive, or ligand-gated. Receptor potentials are graded and their amplitude is proportional to the strength of the stimuli. When a stimulus is strong enough, the membrane potential rises above the threshold of excitation and action potential is produced in the first, the nearest to the receptor, node of Ranvier of a dendrite. Action potentials are not graded. The strength of the stimuli is coded in the frequency of all known action potentials generated. The process in which the energy of a stimulus is converted into an electrical signal in the sensory neuron is termed sensory transduction. Some sensors consist of separate receptor cells associated with terminal endings of sensory nerve fibers by synapse-like contacts. The applied stimuli lead to the opening of stimulus-sensitive channels permitting sodium ions entry that produces receptor potential. This local depolarization opens voltage-gated calcium channels. Calcium ions entry triggers the secretion of neurotransmitters which bind to receptors on dendrite endings, leading to the opening of their ligand-gated channels. Resultant depolarization opens voltage-gated sodium channels and produces action potential in the sensory neuron. Free nerve endings are found in the connective tissue and ramify among the epithelial cells throughout the body. In the skin, they extend into the middle layers of the epidermis. Free nerve endings are sensitive to pain. They are represented by non-myelinated terminal branches of afferent nerve fibers such as medium diameter myelinated A-delta afferents, which form endings detecting acute, well-localized first or fast pain, or small diameter unmyelinated C fibers, which form endings detecting poorly localized second or slow pain. It is also believed that free nerve endings detect touch and temperature. Meissner corpuscles, or tactile corpuscles. They are found beneath the epidermis within dermal papillae with their long axis perpendicular to the surface of the skin, primarily in the glabrous skin. Being concentrated in areas especially sensitive to light touches, such as the fingertips and lips, they are rapidly adapting mechanoreceptors responding to touch and pressure. Meissner corpuscles are oval-shaped, 30 to 150 micrometers in length and 40 to 60 micrometers in diameter. They consist of tightly packed as a stack of pancake, flattened Schwann cells called lamellar cells surrounded by the connective tissue capsule. Up to 6 sensory nerve fibers enter the Meissner corpuscle and meander between lamellar cells. Until recently, it has been believed that all dendrites in Meissner corpuscles are myelinated A-beta fibers. The recent findings have revealed the presence of unmyelinated C nerve fibers, which means that Meissner corpuscles may respond to nociceptive stimuli. Pacinian corpuscles or lamellar corpuscles are located in the dermis and hypodermis of both glabrous and hairy skin, as well as in the interosseous membranes, joint capsules, pancreas and other viscera, breast and genitals. They are rapidly adapting mechanoreceptors that sense deep but not prolonged pressure and vibration. The Pacinian corpuscles are large, up to 4 millimeters in length, and oval-shaped. They possess concentric lamellae separated by spaces containing a network of collagen fibers, some small blood vessels, and clear fluid. The inner core of these lamellae consists of 40 to 80 layers of lamellar Schwann cells. The outer core includes about 30 layers of lamellar connective tissue cells and collagen fibers. Due to their layered structure, the Pacinian corpuscles have an onion-like appearance in cross-sections. On entering the Pacinian corpuscle, the sensory myelinated A-beta nerve fiber loses its myelin sheath and its single dendrite lies in the center of the corpuscle. This dendrite usually gives origin to several branches of nearly equal size, which terminate with end bulbs, or may proceed unbranched and terminate with a spindle-shaped enlargement or an end bulb. Ruffinicorpuscles or bulbous corpuscles are found in the dermis of both glabrous and hairy skin, as well as in joint capsules. They are slowly adapting receptors responding to the stretching of the surrounding connective tissue. Ruffinicorpuscles are fusiform in shape and surrounded by connective tissue capsules. Sensory myelinated A-beta nerve fiber approaching the ruffinicorpuscle loses its myelin sheath and branches, forming dense arborization of unmyelinated dendrite endings encircled by Schwann cells and intertwining with collagen fibers. They exit the pools of the ruffinicorpuscle and interact with surrounding collagen fibers, providing mechanical linking of the inner core of the corpuscle with the surrounding connective tissue. End bulbs of Krause or bulboid corpuscles. They are found in the conjunctiva, in the mucous membrane of the oral cavity, in the synovial membranes of some joints, and in the genitalia, where they are termed genital corpuscles. End bulbs of Krause detect cold, but some authors believe them to sense touch and pressure as well. End bulbs of Krause are oval or cylindrical, consisting of a connective tissue capsule containing Schwann cells and dendrite, which terminates in a coiled-up plexiform mass. To be continued.